The HMPV Virons is pleuromorphic in nature and its size varies from 150 nanometer to 600 nanometer. Hello friends, welcome back to the channel. Like, share, subscribe and don't forget to press the bell icon. The genomic orientation of HMPV resembles other members of the Paramoviridae family. The genome organization of HMPV is quite similar to that of avian pneumovirus AMPV, particularly type C. The genomes of HMPV and HRSV closely resembles each other, excluding a few differences in the order of the genes and the absence of the non-structural genes from the HMPV genome. For HRSV, the two non-structural proteins NS1 and NS2 has been identified as potent multifunctional antagonist of the interferons IFN signaling pathways. The absence of these proteins may be the reason for the difference in the level of host innate immune response observed during HRSV and HMPV infection. The HMPV genome is comprised of negative sense single-stranded RNA and contain 8 genes that code for 9 proteins. The order of the gene in the genome. The proteins are nucleoprotein, N protein, and the phosphoprotein, that is P protein, the matrix protein, M protein, the fusion glycoprotein, that is F protein, the putative transcription factor M21 protein, the RNA synthesis regulatory factor, the M22 protein, the small hydrophobic glycoprotein SH protein, the attachment glycoprotein, that is G protein, and the viral polymerase, that is L protein. The RNA core is surrounded by M protein and covered by a lipase envelope. This envelope contains the 3 surface glycoproteins in the form of spikes of approximately 13 to 17 nanometer. The core nucleic acids are associated with the P, N, L, M21 and M22 proteins and form a nucleocapsid 17 nanometer in diameter. With the help of G and F proteins, HMPV attaches and fuses to heparin sulfate receptors on the cell surface. After the fusion process, the viral nucleocapsid enters into the cytoplasm of the host cell and undergoes replication. The newly synthesized viral genome assembles with the viral P, N, L and M2 proteins and moves towards the host cell membrane. The viron now buds out of the cell with the F, SH and G proteins exposed on the outer side of the membrane. The P protein acts as a cofactor to stabilize the L protein, allowing the formation of the virus ribonucleoprotein complex during the virus replication. The M protein plays a crucial role in virus assembly and budding by interacting with the RNP complex. The N protein encapsulates the viral genome and protects it from nuclease activity. In addition to regulating viral transcriptions and replications, the M2 proteins plays a major role in virulence by decreasing the host innate immunity. Like other members of the Paramyxoviridae family, HMPV interferes with the host innate immune system using specific mechanism. The virus antagonizes cellular responses by regulating pattern recognition receptors such as toll-like receptors and retinoic acid inducible gene-like receptors and other signaling molecules. Infections interferes with dendritic cell activity and reduces antigen specific T cells activation. Virus clearance remain incomplete and the chance of reinfection occurring increases. Members of the two genotypes show much less amino acid and nucleotide similarity than members of the same subgroups, while the same genotype based on the F gene sequence. Comparing all the subgroups, that is A1, A2, B1 and B2, the N gene is found to be most conserved at both the nucleotide and the amino acid level, while the G genes is the least conserved and have 79% and 59.2% respectively. HMPV has been isolated on all continents and have a seasonal distribution. The geographic distribution of the various HMPV genotype outbreaks occur mainly in spring and winter month, January to March in the northern hemisphere and June to July in the southern hemisphere. The peak of the HMPV seasonal case is observed between March and April, following the RSV and influenza infection seasons. The HMPV infections season overlaps with that of RSV infection seasons. Being a respiratory infection, HMPV is transmitted by infectious airborne droplets. Seroprevalence that a high percentage, 90 to 100, of children have been infected by time they are 5 to 10 years old, but reinfection can occur throughout adulthood. This may be due to insufficient immunity acquired during the initial infection and due to infection by different viral genotypes. The incubation period varies from individual to individual, but is commonly between 3 and 5 days during animal experimentation. Peak viral titrates are seen between day 4 and day 5 in mice and cotton rats. HMPV is commonly found in the pediatric populations with high susceptibility rates in children less than 2 year old. HMPV infection in adults normally shows only mild flu like symptoms. However, in some adult cases, especially elder adults, severe complication such as chronic obstructive pulmonary disease COPD can occur. Dysnopia is more likely in adults as compared to children. HMPV infection has also been reported in several immunocompromised patients, such as lung transplant recipients, patients with hematological malignancies, and hematopoietic stem cell transplant recipient. The both genotype of HMPV co-circulated during a typical respiratory virus season and frequent reinfections with different HMPV genotypes occur. Risk factor associated with severe HMPV infections include premature birth, young age, pre-existing nosocomial infection, and underlying chronic pulmonary heart or neural disorders. Investigating the relationship between genotype and disease severity in children have found any significant correlations. Genotype A may be more virulent than genotype B, indicating that is was genotype B that was associated with severe HMPV infections. Compared to HMPV negative children's, HMPV infected children's were found to be more likely to require supplemental oxygen, to have a longer stay in the intensive care unit ICU, and more likely to have undergone chest radiography. About 40% of children hospitalized with HMPV infection were found to have underlying high risk condition like asthma and chronic lung disease. The average annual rate of hospitalizations was about 3 times more in children than that 6-month old compared to children's 6-month to 5-year old. Nosocomial infection has been reported in several modes of transmission. The annual rate of hospitalization due to HMPV infection is equal to that of influenza and parainfluenza 1, 2, and 3 combined, and a recent HMPV outbreak in 2 skilled nursing facilities showed as 11% mortality rate. The severity of disease caused by the discovered virus and the importance of HMPV pathogenesis and vaccine is now becoming clear.